Hello, my name is Adekemi Taylor and I'm a Senior Director of Integrated Drug Development at Certara. I will be presenting the 6th case study about the role of modeling and simulation in the development of Pembrolizumab as an example of model interconnectedness. The case study is based on this paper by De Graaf et al., which gives an overview of the role played by modeling and simulation in bringing the novel immunotherapy Pembrolizumab to patients with melanoma. Pembrolizumab is a programmed cell death receptor 1, or PD-1, blocking antibody developed by Merck & Company for the treatment of various cancers. The initial clinical development focused on melanoma. The clinical development and initial approval were largely based on a single study, Keynote-001, which was a phase 1b first-in-human study of the safety and efficacy of pembrolizumab, first in patients with solid tumors and later in patients with advanced melanoma. The first modeling effort involved population PK modeling and empirical PK/PD modeling of target engagement. The goal was to pick the minimal effective dose for future cohorts using a learn and confirm paradigm. More details can be found at the publication shown here by Elias Schaap et al. The Keynote-001 study consisted of multiple parts. Initial modeling was based on data collected in parts A and A1, which tested doses ranging from 1 to 10 milligrams per kilogram pembrolizumab in a small number of subjects. Pembrolizumab was administered on day 1 and day 28, which means the first 2 doses were separated by a gap of 4 weeks and every 2 weeks thereafter. As shown in the figure on the left, the PK of pembrolizumab appeared to be linear across this dose range. However, it is known that pembrolizumab PK becomes nonlinear at lower doses. Nevertheless, a population PK model was developed for pembrolizumab using this data. The plot on the right shows observed ex vivo interleukin-2 or IL-2 stimulation ratio, and this is the marker of target engagement, plotted against plasma concentrations of pembrolizumab, using circles to represent the observed data points. An inhibitory Emax model was applied to these data as shown by the model predictions represented by the blue curve. Both the data and the model indicated saturation or near saturation of the drug effect at all 3 dose levels. However, there was a large gap in the PK/PD data between the lowest concentrations, which represent trough samples collected 4 weeks after dosing, and the higher concentrations. As a result, the IC50, which is the concentration that produces the half maximal inhibitory drug effect, this IC50 that was estimated was not considered to be reliable, and the PK/PD model was thus not deemed to be robust. We can consider IC50 to represent the potency of a drug, with a lower IC50 meaning higher drug potency because a lower concentration is required to reach the half maximal drug effect. The data collected in parts A and A1 were therefore not considered sufficient to select the lowest dose to use in the larger patient populations in the study. So modeling and simulation using these data were used to guide the design of an additional cohort in the study, cohort A2. The purpose of part A2 was to better characterize the PK and PD of pembrolizumab. Modeling and simulation using the PK and PK/PD models developed from parts A and A1 were used to assess multiple design options for part A2, and the baseline designs are shown here on the left. Simulation scenarios accounted for possible alternative PK/PD properties to those estimated in the models, including unlikely cases. For example, Different drug potencies were applied, with IC50s ranging from 0.02 to 100 milligrams per liter. Simulations also assumed both linear and nonlinear PK. All told, there were 20 different scenarios of PK/PD properties. A detailed description of the exercise can be found in the publication. Designs B and C, which had lower starting doses than Design A, performed similarly and were both predicted to have good performance in adequately quantifying low IC50 values. However, Design C was selected because it was considered to have the more symmetric design, and so Design C was used as the design for Part A2. The design selected for Part A2 was successful in that it filled in the gaps previously present in the data when only data from Parts A and A1 were considered. For example, the newly collected concentration time data suggested concentration-dependent clearance at lower doses, and the population PK model was updated accordingly. Also, as shown on the right, Part A2 was able to fill in the gaps in the PK/PD data, especially at the lower doses, resulting in a more robust PK/PD model. The updated models that included data from Part A2 were then used to simulate target engagement across a range of doses of pembrolizumab. The graph on the left shows the percent target engagement at trough concentrations across a dose range of 0.1 to 20 milligrams per kilograms given every 2 weeks. The graph on the right shows the probability of achieving 95% target engagement, that is, the percentage of subjects predicted to achieve 95% of target engagement at trough, and this was also done across the same dose range. The simulations predicted a high probability of saturating target engagement with every 3-week doses of 2 milligrams per kilogram and higher. 2 milligrams per kilogram has since been tested in larger randomized trials in advanced melanoma and non-small cell lung cancer. This facilitated the approval of pembrolizumab for both indications. Next, we will see how translational PK/PD modeling of tumor growth inhibition in mice was used to support dose selection for pembrolizumab. I will provide a brief overview, and details can be found in the publication by Lindauer et al. That is shown here. The goal here was also to identify the minimal effective dose for pivotal efficacy and safety cohorts. But this time, the exercise used models developed using mouse tumor data and translated them for use in human simulations. Multiple connected model types were used to describe tumor growth inhibition in the mouse. The model was constructed using published physiological parameter values as well as experimental data in tumor-bearing mice, including plasma concentrations of pembrolizumab, receptor occupancy in blood and tumor, and tumor volume measurements over time. There was an empirical PK model for plasma pembrolizumab concentrations shown at the very top, which was a compartmental model describing the distribution and elimination of pembrolizumab as well as reversible binding to the PD-1 target. This was connected to a physiologically based pharmacokinetic or PBPK model that described the concentration of pembrolizumab at the site of action. A semi-mechanistic PK/PD model was used to describe the drug effect at the site of action, with the drug action being related to the drug receptor complex concentration in the tumor. Finally, the tumor growth model described the inhibition of tumor growth by the drug receptor complex, and the size of the tumor at any given time determined the size of the tissue compartment in the PBPK and semi-mechanistic PK/PD models. In this way, the time course of tumor size in the mice could be predicted given a specific dosing regimen of pembrolizumab. The next step was to simulate the dose response in humans, making use of the models developed using the mouse data. The simulated pembrolizumab concentrations were generated using the previously developed population PK model that I described in the first part of this case study. For the pharmacodynamic or PD parameters, scaling was required to translate PD effects from mice to humans. Some PK/PD parameters were considered species invariant, and the values from the mouse PK/PD model were used for these. Other parameters were obtained using a variety of methods, including allometric scaling and in vitro experiments. Multiple scenarios for the rate of tumor growth were considered in order to cover a wide variety of possibilities. So slow, medium, and fast-growing tumors were simulated. The graph on the left shows the tumor size reduction predicted across a range of every 3-week doses of pembrolizumab for the different tumor growth rate scenarios. The other two graphs show the probabilities of different categories of tumor response, namely progressive disease, stable disease, and intermediate, partial, and major response for every 3-week dosing on the left and every 2-week dosing on the right. Both dosing frequencies resulted in similar responses overall, and the probability of achieving partial or major response reached a plateau at 2 milligrams per kilogram every 3 weeks. As you will recall, this was the same dose selected in the first part of the case study, using population PK and empirical PK/PD modeling. Therefore, the 2 milligrams per kilogram every 3-week dose was tested in pivotal cohorts of KEYNOTE-001, in addition to the originally planned dose of 10 milligrams per kilogram. In the next part, we will see how traditional population PK modeling was done to investigate the effect of intrinsic and extrinsic factors on pembrolizumab pharmacokinetics and to support the drug label. This model was based on data pulled from 3 studies, KEYNOTE-001, KEYNOTE-002, and KEYNOTE-003, and included patients with advanced melanoma, non-small cell lung cancer, and other types of tumors. Details of the analysis can be found in the publication shown here by Ahmadi et al. The population PK model was developed using every 2-week and every 3-week doses, ranging from 1 to 10 milligrams per kilogram. A 2-compartment model was used to describe the disposition of pembrolizumab. The graph on the left shows simulated concentration time profiles of selected dosing regimens using the pembrolizumab population PK model. As part of the population PK analysis, the effects of intrinsic and extrinsic factors on pembrolizumab PK parameters was assessed during the covariate search. The statistically significant effects identified on pembrolizumab clearance were sex, estimated glomerular filtration rate, albumin, tumor burden, ipilimumab prior therapy status, and ECOG score. The resulting effects of these covariates on pembrolizumab AUC are depicted on the right. The dashed vertical lines represents the geometric mean ratio for a typical exposure, that is the typical AUC value, which has a value of 1. The circles and error bars represents the geometric mean ratios and corresponding 95% confidence intervals for the covariates at the levels shown. None of the covariates were predicted to result in up to a 0.5-fold reduction or 5-fold increase in the geometric mean ratio of pembrolizumab. Therefore, none were considered clinically relevant, and no dose adjustments are needed for pembrolizumab. In the final part of this case study, we will see how population PK/PD modeling of tumor size dynamics was performed to quantify the efficacy-exposure-response relationship for pembrolizumab, which could be used to interpret the impact of covariates on pembrolizumab exposure. This analysis was based on data in patients with melanoma from Keynote-001, Keynote-002, and Keynote-006 studies, and included ipilimumab-naive patients as well as patients who had previously been treated with ipilimumab. Details of the analysis can be found in the publication shown here by Chatterjee et al. The final model structure used for the tumor size model, which is represented by the equation shown on the left near the top of the slide, can be considered a dose exposure time model. The tumor size at any given time was modeled as a function of baseline tumor size, and a fraction of the tumor, represented by F, was accessible to cell killing by pembrolizumab, and the remaining tumor underwent exponential growth unaffected by the drug. The death rate of the tumor was proportional to drug exposure, and the drug exposure was derived based on the administered dose using the population PK model that was just described in the preceding section of this case study. You'll notice that this is another example of model interconnectedness, with the population PK model providing exposures to be used in the dose exposure time model. Separate exposure response parameters were estimated for the ipilimumab naive and ipilimumab pretreated patients. The set of graphs in the middle shows the model predictions, which are represented as the black lines, overlaid on the observed tumor size data, shown as circles. And the set of graphs on the right display changes from baseline in tumor type over time, with observed data shown at the top and model predicted values shown at the bottom. These two sets of graphs demonstrate that the model was able to capture different types of tumor growth patterns and thus can be considered to be robust. Model-based simulations were then used to predict the rates for different categories of response, specifically disease progression, stable disease, and response. Response being defined as a tumor size reduction of 30% or more from baseline. As seen in the graphs, which show the predicted percentage of subjects in each category for different dose regimens, pembrolizumab exposure was not a significant predictor of response in the ipilimumab experienced or ipilimumab naive patients, as can be seen by similar predicted results across the entire dose range. The results are consistent with those that we showed in the other parts of the case study. 2 milligrams per kilogram every 3 weeks yields maximal or near maximal efficacy of pembrolizumab. With this case study, I hope I have been able to demonstrate to you how different quantitative models were applied at different stages of development for pembrolizumab, and that different modeling and simulation approaches were selected depending on the goals of the analysis and the available data. I showed how information was shared between models as well. The work shown here formed the model-informed drug development framework for pembrolizumab, in which an integrated modeling approach with propagation of information between models was used to support study design for the compound and to support the approval of the 2 milligram per kilogram every 3-week dose, as well as supporting labeling. This pembrolizumab case study provides a demonstration of the importance of model interconnectedness, which was vital to decision-making and facilitated approval of pembrolizumab. This brings us to the end of the presentation.